Welcome to the Dr. Izzy's Medical Podcast channel.
Innate immune system. The innate immune system is one of the two main immunity strategies found in vertebrates, the other being the adaptive immune system. The major functions of the vertebrate innate immune system include recruiting immune cells to sites of infection through the production of chemical factors, including specialized chemical mediators called cytokines, activation of the complement cascade to identify bacteria, activate cells, and promote clearance of antibody complexes or dead cells, identification and removal of foreign substances present in organs, tissues, blood and lymph, by specialized white blood cells, activation of the adaptive immune system through a process known as antigen presentation, acting as a physical and chemical barrier to infectious agents. Anatomical barriers. First one, skin barrier. Defense mechanisms are sweat, desquamation, flushing, organic acids. Second, gastrointestinal tract barrier. Defense mechanisms are peristalsis, gastric acid, bile acids, digestive enzyme, flushing, thiocyanate, defensins, that flora. Third, respiratory airways and lungs barriers. Defense mechanisms are mucociliary escalator, surfactant, defensins. Fourth, nasopharynx barrier. Defense mechanisms are mucus, saliva, lysozyme. Fifth, eyes barrier. Defense mechanism is tears. Sixth, blood-brain barrier. Defense mechanisms are endothelial cells, via passive diffusion, osmosis and active selection. P-glycoprotein, mechanism by which active transportation is mediated. Anatomical barriers include physical, chemical and biological barriers. The epithelial surfaces form a physical barrier that is impermeable to most infectious agents, acting as the first line of defense against invading organisms. Desquamation, shedding, of skin epithelium also helps remove bacteria and other infectious agents that have adhered to the epithelial surfaces. Lack of blood vessels, the inability of the epidermis to retain moisture, and the presence of sebaceous glands in the dermis, produces an environment unsuitable for the survival of microbes. In the gastrointestinal and respiratory tract, movement due to peristalsis or cilia, respectively, helps remove infectious agents. Also, mucus traps infectious agents. The gut flora can prevent the colonization of pathogenic bacteria by secreting toxic substances or by competing with pathogenic bacteria for nutrients or attachment to cell surfaces. The flushing action of tears and saliva helps prevent infection of the eyes and mouth. Inflammation. Inflammation is one of the first responses of the immune system to infection or irritation. Inflammation is stimulated by chemical factors released by injured cells and serves to establish a physical barrier against the spread of infection, and to promote healing of any damaged tissue following the clearance of pathogens. The process of acute inflammation is initiated by cells already present in all tissues, mainly resident macrophages, dendritic cells, histiocytes, Kupffer cells, and mast cells. These cells present receptors contained on the surface or within the cell, named pattern recognition receptors, PRRs, which recognize molecules that are broadly shared by pathogens but distinguishable from host molecules, collectively referred to as pathogen-associated molecular patterns. At the onset of an infection, burn, or other injuries, these cells undergo activation. One of their PRRs recognizes a PAMP, and release inflammatory mediators responsible for the clinical signs of inflammation. Chemical factors produced during inflammation, they are histamine, bradykinin, serotonin, leukotrienes, and prostaglandins, sensitize pain receptors, cause local vasodilation of the blood vessels, and attract phagocytes, especially neutrophils. Neutrophils then trigger other parts of the immune system by releasing factors that summon additional leukocytes and lymphocytes. Cytokines produced by macrophages and other cells of the innate immune system mediate the inflammatory response. These cytokines include TNF, HMGB1, and IL-1. The inflammatory response is characterized by the following symptoms. Redness of the skin, due to locally increased blood circulation. Heat, either increased local temperature, such as a warm feeling around a localized infection, or a systemic fever. Swelling of affected tissues, such as the upper throat during the common cold or joints affected by rheumatoid arthritis. Increased production of mucus, which can cause symptoms like a runny nose or a productive cough. Pain, either local pain, such as painful joints or a sore throat, or affecting the whole body, such as body aches, and possible dysfunction of the organs or tissues involved. Complement system. The complement system is a biochemical cascade of the immune system that helps, or complements, the ability of antibodies to clear pathogens or mark them for destruction by other cells. The cascade is composed of many plasma proteins, synthesized in the liver primarily by hepatocytes. The proteins work together to trigger the recruitment of inflammatory cells, tag pathogens for destruction by other cells by opsonizing, or coating, the surface of the pathogen, form holes in the plasma membrane of the pathogen, resulting in cytolysis of the pathogen cell, causing the death of the pathogen. Rid the body of neutralized antigen-antibody complexes. There are 3 different complement systems: classical, alternative, lectin. Classical starts when antibody binds to bacteria. Alternative starts spontaneously. Lectin starts when lectins bind to mannose on bacteria. White blood cells. All white blood cells, WBCs, are known as leukocytes. Most leukocytes differ from other cells of the body in that they are not tightly associated with a particular organ or tissue, thus, their function is similar to that of independent, single-cell organisms. Most leukocytes are able to move freely and interact with and capture cellular debris, foreign particles, and invading microorganisms, although macrophages, mast cells, and dendritic cells are less mobile. Unlike many other cells in the body, most innate immune leukocytes cannot divide or reproduce on their own, but are the products of multipotent hematopoietic stem cells present in the bone marrow. The innate leukocytes include natural killer cells, mast cells, eosinophils, basophils, and the phagocytic cells include macrophages, neutrophils, and dendritic cells, and function within the immune system by identifying and eliminating pathogens that might cause infection. Mast cells. Mast cells are a type of innate immune cell that resides in connective tissue and in mucous membranes. They are intimately associated with wound healing and defense against pathogens, but are also often associated with allergy and anaphylaxis, serious allergic reactions that can cause death. When activated, mast cells rapidly release characteristic granules, rich in histamine and heparin, along with various hormonal mediators and chemokines, or chemotactic cytokines into the environment. Histamine dilates blood vessels, causing the characteristic signs of inflammation, and recruits neutrophils and macrophages, phagocytes. The word phagocyte literally means eating cell. These are immune cells that engulf, or phagocytose, pathogens or particles. To engulf a particle or pathogen, a phagocyte extends portions of its plasma membrane, wrapping the membrane around the particle until it is enveloped. Once inside the cell, the invading pathogen is contained inside a phagosome, which merges with a lysosome. The lysosome contains enzymes and acids that kill and digest the particle or organism. In general, phagocytes patrol the body searching for pathogens, but are also able to react to a group of highly specialized molecular signals produced by other cells, called cytokines. The phagocytic cells of the immune system include macrophages, neutrophils, and dendritic cells. Phagocytosis of the host's own cells is common as part of regular tissue development and maintenance. When host cells die, either by programmed cell death, also called apoptosis, or by cell injury due to a bacterial or viral infection, phagocytic cells are responsible for their removal from the affected site. By helping to remove dead cells preceding growth and development of new healthy cells, phagocytosis is an important part of the healing process following tissue injury. Macrophages. Macrophages, meaning large eaters, are large phagocytic leukocytes, which are able to move outside of the vascular system by migrating through the walls of capillary vessels and entering the areas between cells in pursuit of invading pathogens. In tissues, organ-specific macrophages are differentiated from phagocytic cells present in the blood called monocytes. Macrophages are the most efficient phagocytes and can phagocytose substantial numbers of bacteria or other cells or microbes. The binding of bacterial molecules to receptors on the surface of a macrophage triggers it to engulf and destroy the bacteria through the generation of a respiratory burst, causing the release of reactive oxygen species. Pathogens also stimulate the macrophage to produce chemokines, which summon other cells to the site of infection. Neutrophils. Neutrophils, along with 2 other cell types, eosinophils and basophils, are known as granulocytes due to the presence of granules in their cytoplasm, or as polymorphonuclear cells due to their distinctive lobed nuclei. Neutrophil granules contain a variety of toxic substances that kill or inhibit growth of bacteria and fungi. Similar to macrophages, neutrophils attack pathogens by activating a respiratory burst. The main products of the neutrophil respiratory burst are strong oxidizing agents including hydrogen peroxide, free oxygen radicals and hypochlorite. Neutrophils are the most abundant type of phagocyte, normally representing 50 to 60% of the total circulating leukocytes, and are usually the first cells to arrive at the site of an infection. The bone marrow of a normal healthy adult produces more than 100 billion neutrophils per day, and more than 10 times that many per day during acute inflammation. Dendritic cells. Dendritic cells are phagocytic cells present in tissues that are in contact with the external environment, mainly the skin, where they are often called Langerhans cells, and the inner mucosal lining of the nose, lungs, stomach, and intestines. They are named for their resemblance to neuronal dendrites, but dendritic cells are not connected to the nervous system. Dendritic cells are very important in the process of antigen presentation, and serve as a link between the innate and adaptive immune systems. Basophils and eosinophils. Basophils and eosinophils are cells related to the neutrophil. When activated by a pathogen encounter, histamine-releasing basophils are important in the defense against parasites and play a role in allergic reactions, such as asthma. Upon activation, eosinophils secrete a range of highly toxic proteins and free radicals that are highly effective in killing parasites, but may also damage tissue during an allergic reaction. Activation and release of toxins by eosinophils are, therefore, tightly regulated to prevent any inappropriate tissue destruction. Natural killer cells. Natural killer cells, short term as NK cells, are a component of the innate immune system that does not directly attack invading microbes. Rather, NK cells destroy compromised host cells, such as tumor cells or virus-infected cells, recognizing such cells by a condition known as missing self. This term describes cells with abnormally low levels of a cell surface marker called MHC1, a situation that can arise in viral infections of host cells. They were named natural killer because of the initial notion that they do not require activation in order to kill cells that are missing self. For many years, it was unclear how NK cell recognized tumor cells and infected cells. It is now known that the MHC makeup on the surface of those cells is altered and the NK cells become activated through recognition of missing self. Normal body cells are not recognized and attacked by NK cells because they express intact self-MHC antigens. Those MHC antigens are recognized by killer cell immunoglobulin receptors that, in essence, put the brakes on NK cells. The NK92 cell line does not express KIR and is developed for tumor therapy. Gamma delta T cells. Like other unconventional T cell subsets bearing invariant T cell receptors, such as CD1d restricted natural killer T cells, gamma delta T cells exhibit characteristics that place them at the border between innate and adaptive immunity. On one hand, gamma delta T cells may be considered a component of adaptive immunity in that they rearrange TCR genes to produce junctional diversity and develop a memory phenotype. However, the various subsets may also be considered part of the innate immune system where a restricted TCR or NK receptors may be used as a pattern recognition receptor. For example, according to this paradigm, large numbers of V-gamma-9 V-delta-2 T cells respond within hours to common molecules produced by microbes, and highly restricted intraepithelial V-delta-1 T cells will respond to stressed epithelial cells. Other vertebrate mechanisms. The coagulation system overlaps with the immune system. Some products of the coagulation system can contribute to the nonspecific defenses by their ability to increase vascular permeability and act as chemotactic agents for phagocytic cells. In addition, some of the products of the coagulation system are directly antimicrobial. For example, beta-lysine, a protein produced by platelets during coagulation, can cause lysis of many gram-positive bacteria by acting as a cationic detergent. Many acute phase proteins of inflammation are involved in the coagulation system. Also, increased levels of lactoferrin and transferrin inhibit bacterial growth by binding iron, an essential nutrient for bacteria. Neural regulation. The innate immune response to infectious and sterile injury is modulated by neural circuits that control cytokine production. The inflammatory reflex is a prototypical neural circuit that controls cytokine production in the spleen. Action potentials transmitted via the vagus nerve to spleen mediate the release of acetylcholine, the neurotransmitter that inhibits cytokine release by interacting with alpha-7 nicotinic acetylcholine receptors expressed on cytokine-producing cells. The motor arc of the inflammatory reflex is termed the cholinergic anti-inflammatory pathway. Immune evasion. Cells of the innate immune system prevent free growth of microorganisms within the body, but many pathogens have evolved mechanisms to evade it. One strategy is intracellular replication, as practiced by Mycobacterium tuberculosis, or wearing a protective capsule, which prevents lysis by complement and by phagocytes, as N. Salmonella. Bacteroides species are normally mutualistic bacteria, making up a substantial portion of the mammalian gastrointestinal flora. Some species like B. fragilis, for example, are opportunistic pathogens, causing infections of the peritoneal cavity and inhibit phagocytosis by affecting the phagocytes receptors used to engulf bacteria. They may also mimic host cells so the immune system does not recognize them as foreign. Staphylococcus aureus inhibits the ability of the phagocyte to respond to chemokine signals. M. tuberculosis, Streptococcus pyogenes, and Bacillus anthracis utilize mechanisms that directly kill the phagocyte. Bacteria and fungi may form complex biofilms, protecting from immune cells and proteins. Biofilms are present in the chronic Pseudomonas aeruginosa and Burkholderia cenecepacia infections characteristic of cystic fibrosis. Viruses. Type 1 interferons, IFN, secreted mainly by dendritic cells, play a central role in antiviral host defense and a cell's antiviral state. Viral components are recognized by different receptors. Toll-like receptors are located in the endosomal membrane and recognize double-stranded RNA, dsRNA, MDA5, and RIG1 receptors are located in the cytoplasm and recognize long dsRNA and phosphate-containing dsRNA respectively. When the cytoplasmic receptors MDA5 and RIG1 recognize a virus, the conformation between the caspase recruitment domain and the card-containing adapter MAVs changes. In parallel, when toll-like receptors in the endocytic compartments recognize a virus, the activation of the adapter protein TRIFE is induced. Both pathways converge in the recruitment and activation of the IKK-epsilon-TBK1 complex, inducing dimerization of transcription factors IRF3 and IRF7, which are translocated in the nucleus, where they induce IFN production with the presence of a particular transcription factor and activate transcription factor 2. IFN is secreted through secretory vesicles, where it can activate receptors on both the same cell it was released from, also known autocrine, or nearby cells, also known paracrine. This induces hundreds of interferon-stimulated genes to be expressed. This leads to antiviral protein production, such as protein kinase R, which inhibits viral protein synthesis, or the 2,5 oligodenylate synthetase family, which degrades viral RNA. Some viruses evade this by producing molecules which interfere with IFN production. For example, the influenza virus produces S1 protein, which can bind to host and viral RNA, interact with immune signaling proteins or block their activation by ubiquitination, thus inhibiting type 1 IFN production. Influenza A also blocks protein kinase R activation and establishment of the antiviral state. The dengue virus also inhibits type 1 IFN production by blocking IRF3 phosphorylation using NS2B3 protease complex.
Thank you for listening Dr. Izzy's medical podcast series. If you want to support this project, please subscribe the channel and like this video. Thank you.